routine day after day that we have. So it was different, and I had a lot of fun preparing.
Oh, that's great. Thank you very much for accepting our invitation. We are honored that you prepared for us.
I'm pleased to collaborate with such wonderful people. Thank you.
Thank you very much.
Thank you. Can you wait 2 minutes?
Sure.
We usually start at 4:04.
Would you like to share your screen to test your presentation, Dr. Modlin?
Sure. Let me move from the Hollywood sign to my office, which I'm not really in, but that's okay.
Yeah.
I could feel like I'm in it. I'm at home.
It's okay.
How's that? That work okay?
Yeah.
Okay. I will be in the chat with Thiago. And Paula, please, can you introduce? Okay, and
okay.
You have any thoughts? Okay.
Good afternoon, everyone. It is our pleasure to introduce you to Dr. Robert Modling, that will talk to us today about antimicrobial pathways in leprosy. We first would like to thank you, Dr. Modling, for accepting our invitation, and I'm going to invite Tiago Calvo to present you, to introduce you to the students. Thank you very much.
Hello, everyone. Good afternoon. My name is Thiago. It is with great honor that I'm going to talk about Dr. Modling a little bit. So Dr. Modling completed his undergraduate studies at Johns Hopkins University. He then received his medical degree at the New York University School of Medicine, and then was followed by a pediatrics intern at the same place. He then pursued a dermatology residency at Los Angeles County University of Southern California School of Medicine. And since the 90s, Dr. Modling has been at UCLA, University of California, Los Angeles, presently as the Klein Professor of Dermatology, Professor of Microbiology, Immunology, and Molecular Genetics. And he's also the chief of the Division of Dermatology and Vice Chair for Cutaneous Medicine and Dermatologic Research in the Department of Medicine. Dr. Modling's research interest, especially in leprosy, began during his dermatology residency at the University of Southern California Medical Center, learning from Dr. Thomas Ray. The goal of his research is centered around the study of leprosy as a model to learn about mechanisms of host defense in humans. As such, Dr. Modling laboratory has made fundamental insights, especially into T-cell subsets, cytokine patterns, antigen presentation, innate immunity, and antimicrobial mechanisms in the immune response to infection. So far, he has published over 180 articles, including 13 papers in Science and Nature and 4 in Nature Medicine. Modling's lab manuscript identifying distinct T-cell cytokine profiles in leprosy has been cited over 1,000 times. An exciting recent discovery is the elucidation of the mechanism by which vitamin D subsets contributes to innate immunity against tuberculosis in humans. Dr. Modling received several awards, including the Montana Award for Outstanding Young Investigator from the Society for Investigative dermatology and the Merit Award from the NIH for his research on toll-like receptors. He has served as a deputy editor for the Journal of Immunology, as well as being a member of the editorial boards of several journals ranging from fields like clinical investigation to infection and immunology. With enormous pleasure, please help me welcome our speaker today, Dr. Robert Modling.
Thank you very much. That was a fantastic introduction. I'm not sure I deserve that, but I thank you anyway. And I'm honored to be here and talk to you. Now, I decided that given the pandemic, people need some excitement. And for me, that wasn't going to be to review some of the major accomplishments that our lab had. So I felt like I had to tell you something new. And so what will do in the first 3 slides or 4 slides, review the first 27 years of my research. And then after that, all the slides are new data that began when I visited Brazil last time, which was in November of 2017. So I will try to go slowly, but please stop me if I'm unclear or if you have questions or I'm going too fast. I'll do my best. So like many of you, I've been fascinated since I was a medical student and then as a dermatology resident by the spectrum of leprosy, because its diverse clinical manifestations correlate with the level of the immune response to the pathogen Mycobacterium leprae. And so tuberculoid patients represent resistance to infection. They have few skin lesions, few bacilli, and strong cell-mediated immunity, and can actually, over time, eliminate the pathogen. Whereas lepromatous patients are unable to control the infection. They have many skin lesions, numerous bacilli, a small amount of cell-mediated immunity, mainly antibody responses, which are not protective. And within this are reversal reactions, patients that typically start towards the lepromatous pole and upgrade towards the tuberculoid pole, so providing an opportunity to study the dynamics of the immune response. So this is a typical patient with tuberculoid leprosy, a single skin lesion with few or no bacilli, and patients with lepromatous leprosy with disseminated infection and a massive number of bacilli and macrophages. So this we all know and are interested in. So it was in December 1981 when I was a dermatology resident that monoclonal antibodies had just been invented, and professor at USC had learned immunoperoxidase, and so we looked to see what the T cell subsets were in leprosy lesions, and I looked through the microscope and saw the following: that in the granulomas, which collections of lymphocytes and macrophages in tuberculoid leprosy, there were CD4 cells throughout, whereas the CD8 cells formed rings around the granuloma. Lepromatous leprosy, there were more CD8s than CD4s, and these were mixed together. And so it was fascinating to see this pattern of the granuloma architecture. And that is probably a major reason that I decided to continue in science, because this discovery was interesting and exciting, then go into clinical dermatology. And so I've been pursuing that one day I would hope to understand the architecture of granulomas. That day hasn't come yet, but we're getting closer. So these are then in tuberculoid leprosy or reversal reactions. The granulomas are somewhat organized with a core of macrophages, a mantle zone of T cells, but this is the classic view that granulomas are a collection of macrophages and lymphocytes. But as we know now, some of the T cells are sprinkled throughout, whereas in lepromatous leprosy, there are less mature macrophages, histiocytes, and a sprinkling of T cells. So from 1984 to present, we've been interested in the antimicrobial pathways that lead to killing M. leprae and other mycobacteria. And I think there are three major areas that we've studied, and these are available. These are published, and so you can read about them in more detail if you'd like. So the first was, as mentioned, defining how vitamin D contributed to an antimicrobial response, and we showed that through toll, ligand, or interferon gamma activation. There was induction of IL-15, which induced an enzyme, CYP27B1, a cytochrome P450 enzyme that catabolized the conversion of 25 hydroxy vitamin D to 125 dihydroxy vitamin D. There was upregulation of the vitamin D receptor, and that led to the induction of two antimicrobial proteins, cathelicidin and DEFB was the identification that T cells expressed an antimicrobial protein, granulysin. And I just noticed my internet was a little unstable, so I apologize. My mother-in-law has been living with us for a year. When she microwaves her tea, that happens. I haven't been able to figure that out. So anyway, so granulysin was expressed by T cells, and subsequently we showed that the combination of granzyme perforin and granulysin led to an effective antimicrobial activity against bacteria and macrophages. And so these cells were antimicrobial CTLs, but by having granzyme perforin and granulysin, we term them tricytotoxic T cells. And these cells also express NK markers, which can activate them. And the third pathway was the finding that Th17 cells expressed IL-26. And this complexed with DNA entered infected macrophages, activated STING to induce autophagy. IL-26 also co-localized with the bacteria through autophagolysosomal fusion, and this led to an antimicrobial response against the bacteria. So we had investigated three distinct antimicrobial mechanisms by which the host immune response could kill intracellular mycobacteria. And it's interesting that none of these pathways are present in mice. The mouse cathelicidin gene is not regulated by vitamin D. Granulysin is not present in mice, and neither is IL-26. So what I realized was that over these years, I lost sight of my original interest was the architecture of the granuloma. And how does the architecture of the granuloma contribute to host defense? So that started the studies beginning in 2017, coincidence with my last trip to Brazil to study the relationship of antimicrobial pathways to granuloma architecture. And although it's been said that the granuloma is a collection of macrophages and lymphocytes, skin is not a collection of macrophages and lymphocytes. It's a complex structure with epidermis composed of layers of keratinocytes and Langerhans cells, which are antigen-presenting cells specific to skin. And in the dermal layers, there are structures such as sebaceous glands, hair follicles, blood vessels, nerves, and cell components include fibroblasts, macrophages, T cells, mast cells, and K cells. So November 27th, 2017, we came to Rio, and Travis Hughes is an MSTP student at MIT who we've collaborated with, and he came to teach us how to do single cell sequencing of leprosy tissues. And we looked under the microscope, and he was able to seed single cells into wells containing beads. And so this was, we were excited and celebrated with great food. Patrick Brennan was there at the time as well, and some of the members of my lab, David Weiss, Bruno, and Priscilla. So this is single cell RNA-seq using seqWell. So we take a skin biopsy sample, and in it are the granulomas and other cells, and we dissociate this into a single cell suspension. And this is added to a glass slide containing microwells, and these have beads in them. And so by using seeding according to Poisson distribution, you end up with single cells per well, mostly. And in those wells are barcodes, are beads that have barcodes that identify that particular cell. And then the library is made. These cells can be mixed together, and you end up with data which can be analyzed, which I'll explain as we go forward. So this is what the data looks like. This is Seurat uses UMAP, which is kind of similar to PCA analysis, but for large data sets. And so these first two dimensions show the differential nature of each individual cell. And these cells then form clusters, and we can identify these clusters by marker genes as T cell, Langerhans cell, myeloid, plasma cell, keratinocytes, et cetera. And in this part of the diagram, I'm showing you now in red, which will be the manner which will show future slides. Red is Lepromidis leprosy, because the bacteria are phyte positive, so that's why they're red. And reversal reactions are blue. And so you can see that there are some cell types, like in, for example, the T cell, there are areas that are enriched for reversal and those that are enriched for Lepromidis leprosy. We see that in the myeloid populations as well. So this gave us, was exciting that we could obtain cells from lesions, and these are 5 reversal reaction and 5 Lepromidis lesions, and we could identify cell types and see at the first level that there are some differences between reversal reactions and Lepromidis leprosy. Now, although we used oligo DT to select for poly A transcripts, we're able to detect some mLepre reads, because there are some ribosomal regions that actually have are rich for adenosine, even though it's a high GC containing bacteria. And you can see that we could detect them Leprosy reads in all of the Lepromatous patients and one of the reversal reaction. So now this shows, you'll see a series of diagrams like this that show for each cell population what the contribution was for the 10 different specimens. And so what you'll notice from this is first that the 10 different specimens are mostly contributing to each cell type. And for most of the cell types, there's a contribution from Lepromatous and reversal. And there are two exceptions. One is plasma cells, which were mostly detected in Lepromatous leprosy in red here. And then Langerhans cells, which we detected in 3 reversal reaction patients. And that was because we immunoselected for CD1A Langerhans cells in those individuals. Now, this is a heat map for the different clusters, and this shows you at one level how we identify what the cell population is. So the T cells have CD3D, CD3E, T cell receptor beta. B cells have CD79A. The plasma cells have immunoglobulin transcripts. Myeloid cells have lysozyme. The Langerhans cells have CD1A and CD207. The keratinocytes have keratins. Fibroblasts have collagens. The endothelial cells have PCAM. The eccrine glands and melanocytes, tyrosinases. So this is how we're able to identify what these different cell types are. So we were then able to take those cell types, for example, T cells, and subcluster them to see what, if there were different populations. So we saw 7 different T cell subtypes because we always start numbering from 0. So TC0 here expressed RORC and IL-23 receptor, and these are Th17 cells. And we don't really see much IL-17 because of the dropout with this sequencing and the low expression copy number of some of those cytokines. But these genes are characteristic. Cluster 2 are here are CTL, and we call them RRCTL because they're mostly in reversal reaction lesions. And they're more immature. They express interferon and granzyme K. And TC2 are CTL that are mostly in Lepromatous lesions. And these are some of the genes we saw were genes typically downstream of type 1 interferon. TC3, which is here, are central memory T cells. And then this is followed by naive stress cells. These are, they express Jun, N4, NR4A1, FOS. These are clearly T regulatory cells with the typical markers. And these cells express granzyme perforin granulysin. So those are tricytotoxic T cells or antimicrobial CTL. So this looks really simple, but actually I just wanted to show you that you get the data like this with lists, and then you start hunting around for genes. And I've done this thousands of times now. And so this is how you identify various genes then that tell you what the cell types are. But there are plenty of other genes that you have no idea what they are, and they may provide insight into these cells, and certainly be interested to study them. So again, I want to show you now feature plots to show you the distribution of some interesting genes. So this is interferon gamma. And so positive cells are in red here. And you can see that they're expressed in TC2 over here. They're also in TC1, so the different CTL populations, and TC6. And in the Th17s, they express, TC0 express that. So there's the Th17s, RORC, and you can see that overlaps with the interferon gamma. Granzyme B, the different CTLs, overlaps with interferon gamma, perforin. And granulysin is predominantly expressed in cluster 6, which are the tri-cytotoxic cells. And here's FOXP3 and the T regulatory cells. So you can identify the specificity of the marker genes for different clusters. So I'm going to focus now on two things. What is the difference between these RR and LF-CTL, and also in comparison to the CTL that we call antimicrobial CTL. So this, first to show you the distribution of the cell types. So TC0, which are the Th17 cells, are mostly in reversal reactions. RR-CTL, about 80% reversal reaction. LF-CTL, 90% Lepromidis. Central memory, these naive stress, and Tregs, surprisingly to me, they're in both. And then these antimicrobial CTL look like they're in both as well. So if you start seeing interferon genes and you want to know if there's a difference as to whether they're type 1 or type 2 interferon, there's only one person in the world that I know to go to, and that's Rosanny. She established her expertise with her science paper in 2013, looking at type 1 and type 2 interferon inducible genes in leprosy. And so she applied her algorithms to these different CTL, and you can see that the reversal reaction CTL had equal numbers of enrichment of type 1 and type 2 interferon genes, but this was not significant. And then the Lepromidis CTL had a 3.5-fold enrichment for type 1 interferon genes, which was highly significant. So the difference between the CTL populations in reversal reaction and Lepromidis is that the Lepromidis CTL have been apparently exposed to type 1 interferons, regulate their type 1 interferon downstream genes. Now, we know type 1 interferons required for induction of CTL, but what the long-term exposure of type 1 interferon is on CTL function is not known. And so this prompts a new area of investigation. Now, to focus on TC6, what we noticed is, although they were present equally in Lepromidis and reversal reaction lesions, there was a distinct distribution of the tri-cytotoxic genes. So granzyme B was much higher in reversal reaction, perforin was much higher in reversal reaction, and granulysin was maybe half a log higher. And if you score this together, the score was higher in reversal reaction. So within this cluster, TC6, there was a difference in the cells from reversal versus Lepromidis, suggesting that the tri-cytotoxic cells were enriched in reversal reaction lesions. Now, another thing we looked at was the presence of interferon gamma. So there are about 200 total CTL that were detected in either reversal reaction in RRCTL or the antimicrobial CTL. And there were more cells in the reversal reaction cluster. The antimicrobial cluster was smaller. And in blue here, you see the number of interferon gamma expressing cells, and it's similar to the number of granzyme B expressing cells, and also the perforin expressing cells. However, the number of interferon gamma cells is far greater in the reverse reaction CTL than the number of granulysin cells. Well, most of the granulysin cells express interferon gamma as well. So interferon gamma is not a marker for these antimicrobial CTL. It's actually a marker mainly for CTL that don't express granulysin. And this shows in more detail how this looks. So TC1 is here, and TC6 is here, and interferon gamma only is without granulysin is here, and interferon gamma and granulysin is mostly in this cluster 6. Whereas if you look at granzyme B and perforin, you see that there are double positives in both areas. So what this means is that in summary, RRCTL were more abundant than antimicrobial CTL, such that detection of interferon gamma is an appropriate surrogate measure of all CTL, but not of the specialized antimicrobial CTL, which require expression of granzyme, perforin, and granulysin for their antimicrobial activity. So these more immature CTL, such as RRCTL, have the potential to kill infected macrophages, but they don't release granulysin, so the killing of the cell would release viable bacilli, resulting in disseminating the intracellular bacteria rather than controlling the infection. And in human vaccine trials for tuberculosis, there is a critical need for correlates and biomarkers of protection, and that just measurement of interferon gamma production by CD8 cells, which is what's happening, would simply not serve as a measure of antimicrobial CTL, which would be directly estimated by measurement of granulysin or specific surface marker correlates. So before I move on to myeloid cells, I don't know if there are any questions. I don't see any in the chat. So if anybody wants to speak up, they're welcome to. If not, let's move on to the myeloid cells. So there were 5 populations of myeloid cells, and this is 0, ML0, myeloid 0. And you can see it as CLEC10A and CD1C. So this is a myeloid dendritic cell. And then there's this cluster 1, which again has type 1 interferon genes. So we call this a type 1 interferon macrophage. And then there was this cluster 2, which took me a long time to figure out. It expressed sort of a mixture of M1 and M2 genes. And ultimately, we recognized that they expressed TREM2, and we call them TREM2 macrophages, and more about those later. Anna Paula wrote, we have been seeing that in tuberculosis patients that present higher bacillary load, the CTL reduces the amount of perforin and granzyme. Is there any correlation with RNL patients' responses? What do you think about Th17 comparing tuberculoid and lepromatous lesions? Do you believe that the Th17 are final developed? Okay, those are 2 excellent questions. So I'm not seeing a reduction in if you go back to this list here, and you look at CTLR, they have granzyme B and perforin 1, and well, CTLL have them, but I think they're further down on the list. And I really couldn't say that the expression was lower. So I wonder if the type 1 interferon genes may affect their function. But I was not able to say that there was a decrease. The main difference was, although if you look at that population, TC6, there seemed to be, which was a smaller population, there was clearly a lower amount of granzyme and perforin. And this is the one that counts because it has granulysin, the antimicrobial protein. So I would say that yes, there's a reduction of granzyme and perforin, but only in the tricytotoxic cells that express granulysin. And the other question was related to Th17 differentiation. Well, those cells were only present in reversal reaction lesions, and I can't say anything about the differentiation. We've done some samples by 10x, which is a little more sensitive for low expressing genes, and we're able to detect IL-17, and I'll show you some other genes later. But it's true, the levels are not like are seen in psoriasis Th17s. So whether that represents incomplete differentiation or lower activation, I don't know. I think it's an interesting question. Okay, so we're back. We just talked about these TREM2 macrophages. Now this is really interesting. Cluster 3 has this. So this was really exciting. So when we saw this is really weird to see this trail of cells, cluster 3, and they sort of have a mixture of genes that are in cluster 2 and 4, and so we call them transitional. While cluster 4 is it has M1-like genes, so these are M1 macrophages. So this is reminded me of when in school, if we were going to from the classroom to the auditorium, we had to line up in single file and march our way from one place to another. And so it looked like these cells were kind of going from here to here. And so we'll come back to that later, what the meaning of this single file is. And you could see here now that the dendritic cells are mostly in reversal reaction. The interferon-like macrophages in Lepromidis, the TREM2 macrophages in Lepromidis, these transitional macrophages are a mix, and the M1 macrophages are in reversal reaction. So that's what I just went over in a different way. And here's the TREM2 macrophages. TREM2 is expressed higher in this cluster 2, lower in cluster 3. ApoE is a marker, and then it goes down. And if you look at a module of TREM9 genes, it goes up as you get to cluster 2 and then down as you move to cluster 4. So TREM2 macrophages, and that might be a future talk, have been identified in atherosclerosis, Alzheimer's, adipose tissue, fatty, necrosis, non-alcoholic steatohepatitis, for example. And they're probably the foam cells in leprosy and tuberculosis. They have a large number of lipid metabolism genes, and but they also express inflammatory genes. So they interconnect lipid metabolism with inflammation, and and there are a number of papers about them, as I've mentioned, in different diseases in which foamy macrophages are present. So for keratinocytes, we also saw that there were differences in reversal and lepromatous. For fibroblasts, we saw populations here and here that were only in reversal, and 2 that were in lepromatous. And endothelial cells, there were some specific for reversal reaction, and these were present in lepromatous lesions. So now that we have all these cell types and subtypes, given that leprosy reversal reactions are associated with a reduction in viable M. leprae bacilli lesions, we sought to determine the array of antimicrobial genes that were present in defined cell populations. And to do this, we used the Z-score, which I thought I'd explain in case people don't know about it. So the Z-score is calculated by taking a specific number you're interested in, subtracting the mean, and dividing it by the standard deviation. So here is a hypothetical expression levels of genes in various cell types. So let's say for this gene, in reversal reaction in TC0, it's 8. While in lepromatous cells, even though there are a few of them, it's low, it's 1, and so on. I've picked some various cell types. We do this in all the subtypes, but for the purpose of this, it was easier to, I've just picked these. And then, so we calculate the mean, which is 2.9, the standard deviation 7.5. And now we can enter 8 into the formula, and the Z-score is 3.6. And for the other ones, the Z-score is negative. So what does this accomplish? What it accomplishes is that if you look at the raw score of a set of data, it could range with a mean of 4 and the standard deviation up to 7 down to 2. And for another gene, it could go from 20 to 30. What the Z-score does is it normalizes everything around 0 with a standard deviation of 1. So now you can compare the expression of one gene to another relatively. It's the relative expression of that gene across all clusters. So from 21,318 cells from 10 biopsy specimens, there was an average of 741 genes and 3,556 transcripts per cell. 43,000 unique genes were detected. We took a list. Priscilla is the expert on antimicrobial genes. 1,404 antimicrobial genes were identified in gene cards, and of those, 1,124 had a Z-score greater than 3 in reversal reaction versus lepromatous in a particular subcluster. And so this is what the Z-score looks like now for IL-26, a Th17 cell gene. So you can see here, there are some IL-26 positive cells, maybe 30 or 40. And this is in, and that's the only cluster that it's above 3. And I just, I wasn't sure you could read this, so I amplified it, and that's in TC0. The Th17 cells express IL-26. So that's an example of the Z-scores. And we looked at the expression of the Z-scores of these 1,124 antimicrobial genes, and they're higher in reversal reaction compared to Leprosy leprosy. And Rosani looks at the upstream regulators and found they were regulated by interferon gamma, TNF, IL-1, and the expression of those regulators is also higher in reversal reaction. So then the next aspect of the analysis was something called pseudotime. And when I heard about pseudotime, I was so excited. How could something be called pseudotime? Well, now from the pandemic, I think we have a feeling of what pseudotime may be. But actually, it's an informatics term. But immediately, I thought about when I was a teenager and younger, going to the Museum of Modern Art in New York. And so I began to think, what is pseudotime? And there was this painting I used to love, The Persistence of Memory by Salvador Dali. So is this pseudotime where this is what the clocks look like? Or my favorite painting of all time is called Broadway Boogie Woogie by Pierre Mondrian. And he's showing the streets in New York in a way with these colors that it looks like there's actually movement. So is that pseudotime? But that's not what pseudotime is. So what it is that let's say that you had a collection of cells that were stimulated or differentiating from here to here over time. And you capture them and do single cell analysis, and now they're all mixed up. You've lost the time information. It's not barcoded for time. So what pseudotime does, it statistically infers the ordering of the cells. So if you start with this cell, what's the cell that's most like it? And what's the cell next most like it, et cetera. So you end up progressing in a linear way from here to here. So genes are differentially expressed over time in this model. So the same way, when we look at that single cell file here from 2 to 3, 2 to 4, we can order the cells in 2, 3, and 4 in what is a sequential way. And this also moves from Leprosy to reversal. And in this are various clusters that start with what we call patterns. Pattern A is higher at the beginning, then this is pattern B, C, D, E, F. So you can see the patterns are evolving. And you can see here that I've marked 2 upstream regulators, IL-1 and interferon gamma, are involved in this pattern. Same for keratinocytes. We saw a similar type shape, and these could be ordered into a pseudotime. And we show it from Leprosy to reversal. Now, the pseudotime doesn't tell us where it starts and ends, but we know clinically, in most cases, it goes from Leprosy to reversal. And it went through these clusters, and there's a progression of genes, which are also regulated by IL-1 and interferon gamma. So if we looked at the upstream regulators for this keratinocyte and macrophage pseudotime, which we think may represent differentiation or activation, you can see that IL-1 and interferon gamma were 2 of the top upstream regulators. And you can see there's a progression of the targets. This is for the macrophages. The keratinocytes are less regulated by interferon gamma, more by IL-1. So these are the upstream regulators that were important for the antimicrobial genes and the pseudotime IL-1, which was expressed in Langerhans cells, which Bruno is our expert on, and dendritic cells. Interferon gamma was in TC0 and TC1. And we could connect this to the antimicrobial genes and various cell types that were regulated by this. So IL-1 on the left here would induce this set of genes in T cells, including IL-26, and these genes in fibroblasts. And very few genes are shown in the Lepromidis clusters or in the clusters that are mixed. Interferon gamma is produced here and has effects on various cell types, mostly ones in reversal reaction. And this is a compilation of some of those antimicrobial genes by Z-score. So these are expressed in myeloid cells and Langerhans cells and in M1 macrophages mostly. These are in fibroblasts, and these are some keratinocyte-specific antimicrobial genes. These are some of the Th17 genes. And notice, again, IL-17 is not here, but some other more antimicrobial genes were characteristic. And then here's granzyme perforin and granulysin in the TC6 over here. So we then, from these 1,124 antimicrobial genes, we now focused on 66. And that was based on the fact that 59 have been shown to contribute to the immune response to mycobacterial infection, either expressed at the site of infection, induced by mycobacteria, or have mycobacterial activity. And 41 of those have been shown to have direct antibacterial activity from a database. So of the 66, there were 7 that have not been shown to be involved in mycobacterial infection, but were directly antimicrobial. So out of the 1124, only 41 have been shown to have direct antibacterial activity. And now this is a heat map showing the Z-scores of those 66 antimicrobial genes. And what you can see here is that there were a set of antimicrobial genes in endothelial cells, a set in T cells, a set in keratinocytes, a set in myeloid cells, and a set in fibroblasts. And although there's a little bit of spillover, some genes like this fibroblast gene was also found in a myeloid cell, it seems that there are cell-specific patterns of antimicrobial gene expression. So then we could put together a diagram then that there are various cell types that express antimicrobial genes that may contribute to antibacterial activity in the macrophage. And I drew this picture of a granuloma, but there's no architecture, it's just a cartoon. And so in a way, it's disappointing that we know what the cells in the lesions do, but we don't know where they are. So this is data from just the past couple of months, working with Johan Gudnason in Michigan, and all the work I've shown, the informatics work was done by Fei-Young Ma, except for certain examples that I alluded to that were done by Priscilla, Rosani, and Bruno. So now what's possible is you could take a slide, and on that slide, you put a section of tissue on the slide. And you can fix the tissue, you do an H&E stain, and then within each of these little chambers are barcoded spots that after permeabilization capture the material from the cells. And then you do cDNA synthesis and you do a library. So this is what it looks like here. You put your tissue here. There are these different spots, and the spots are 55 micrometers in diameter, and there's some space between the spots. You capture, the blue captures the cells in that well, the RNA. This captures from that well, and they're barcoded. So here, this one has the green barcode, and so now after you make the library, you know the location it came from on the plate. So this is unbelievable. So this is a tuberculoid leprosy lesion, and I noticed my favorite thing here. There's macrophages, and then there's a ring of lymphocytes around it that actually kind of extends through here. But here, within here is the classic organized granuloma. And so this now shows you what the spots look like on top of this. And this is the actual section from which the RNA is extracted. So it's a frozen section. So, but this is the data I'm going to show you are from this exact specimen that's 20 micrometers in thickness. And so you can see now, one issue is that the spots contain a collection of cells that can range from 10 to 30 cells, depending on the diameter. But we do have spots that are in the T cell rich areas and those in the macrophage rich areas. So this data can be analyzed, and this is how Fei-Young analyzed it. So he took the single cell RNA-seq data and the different signatures for the cell types and interrogated each spot, the sequencing data from each spot. And then I contributed to this idea. We represented each spot as a pie chart, okay? And so I've amplified this pie chart here. So you can see here's a pink one. Well, this means that all the cells were myeloid. But if you see this one here, so this is about, I don't know, 80% myeloid, 80% T cell and 20% myeloid. So the scatter pie chart shows what is in each spot in terms of, okay, it looks like 80% of the genes are from T cells and 20% from myeloid. And some are more complex. But largely we can define spots that are collections of just predominantly one type of cell. So we now take this scatter pie deconvolution and assign cell types by k-means clustering, and we're able to assign endothelial, T cell, keratinocytes, myeloid, fibroblasts. And the gray ones are spots that were too complex, we couldn't define. One other point is that we did look at the Langerhans cell signature, and you could see some keratinocytes had a small number of Langerhans cells, but we're in purple here. But there weren't, the resolution is not sufficient to really look at Langerhans cell homogeneous populations of Langerhans cells. We also looked at a reversal reaction lesion. You could see the granulomas, and you could see that we could assign certain areas, pink is myeloid, that were the aggregates of myeloid cells and surrounded by T cells, and green is fibroblasts. And we looked at some of the antimicrobial genes of interest, and you can see one was CYBB, which we found specifically expressed in the myeloid cells. And you could see that its expression was in the areas of myeloid cells. CCL5 was expressed by T cells, RANTES, and it's in some of these blue areas that are T cell rich areas. MMP2 was in fibroblasts, and it's in some of these green areas where the fibroblasts are. And KLK5, which encodes a gene that, an enzyme that cleaves cathelicidin, was present in the epidermis. So this is a reversal reaction. This is the entire tuberculoid leprosy biopsy, and again, we can identify the location of these 4 classic genes in the different cell populations. So we, out of the 66 antimicrobial genes that we detected by the single cell RNA-seq, 55 were detected by the spatial seq, and 35 were detected in the cell clusters that we felt were similar. So you could see that this validates then the single cell data, that the genes expressed in the endothelial cells by single cell RNA-seq were expressed in the endothelial cells as a whole. It was not as easy to subtype them, you'll see later. T cell genes were expressed in the T cells, keratinocyte genes in the keratinocytes from reversal in the borderline tuberculoid, myeloid cells in the myeloid populations of fibroblasts equivalent as well. So this validates the single cell data, but with spatial seq data that tells us the location. So these are the different subpopulations. And so what's interesting is here's fibroblast 0, and it's identified by red. And we used a signature now for fibroblast 0 to look at where the fibroblast 0 is located. And it's mostly in the superficial dermis, where fibroblast 2 is around the granulomas. Keratinocyte, there are various populations. Myeloid 3 and 4, the transitional and M1-like, are in the myeloid aggregates, and there are a few ML0 dendritic cells around them. And this is the tuberculoid that shows a very similar pattern. Fibroblast 2 is present, 0 is up in the part of the epidermis here, and again, fibroblast 2 is near the granulomas. ML0, the dendritic cells, are around the granulomas, and ML3 and 4 comprise these granulomas here. Well, to really understand the architecture, I focused on that region that I told you I was interested in previously. And we looked at the scatter pie and the cell types, and you could see that we do have this core of macrophages, this ring of T cells, and some fibroblasts surrounding it. And from this, we're able to now, the gene expression data from this region, we are now able to put together this diagram of the architecture of the granuloma. And I'm very proud that I actually drew this in BioRender. What it shows now is the various cell types we've identified by single cell and spatial seq, and the antimicrobial genes they express, the 66 antimicrobial genes. So the Langerhans cells have a certain set of antimicrobial genes, and there are different antimicrobial genes in different layers of the epidermis. And the granuloma itself is has a core of the transitional ML3 and M1-like ML4 macrophages that have their own antimicrobial genes, including some of the vitamin D pathway. There's also a mantle zone of T cells that include the Th17s, CTL and the antimicrobial CTL that at present we detect mostly around the periphery. There's fibroblasts, and there's fibroblast 2 that have a number of antimicrobial genes that are also inflammatory. And there are fibroblasts located here that have an overlapping but distinct set of inflammatory genes. The dendritic cells are located near the T cells, and they also have antimicrobial genes, and then they're scattered endothelial cells. So then the granuloma is not just the granuloma. The granuloma has its own architecture, but there's also cells in the surrounding tissue, the skin, that likely contribute to the immune, and in particular, what we're interested in, the antimicrobial response. So this paper has just been submitted, all the data I showed you today, and I'm grateful to the collaborators, many of which I mentioned as we went through today. And in summary, we identified differences in the distribution of specific cell types and subtypes between 2 distinct clinical and immunologic forms of leprosy, reversal reaction, and Lepromatosis leprosy. We constructed pseudotime trajectories for macrophage and keratinocytes reflecting progression of cells from Lepromatosis leprosy to reversal reaction. We constructed antimicrobial gene networks preferentially associated with host defense in leprosy, regulated by IL-1 and interferon gamma. We identified the distinct spatial locations of cell types and antimicrobial genes in and around the organized granuloma and reversal reaction and tuberculoid leprosy. The granuloma is not limited to an organized core of macrophages with lymphocytes, but extends to other cell types such as keratinocytes and fibroblasts to combat the foreign invader. And so in summary, it takes a village of cells to kill the bacteria. So I want to thank you very much for, again, the invitation. And I'm so excited to present this new data for the first time. And I'm happy to answer any questions.
Thank you very much. It was overwhelming. I love all the results, and it's impossible to do that in Brazil.
Well, we did some of it in Brazil, actually. So I don't know, we did it. Although we had a lot of trouble doing it because of all the specimens we processed in Brazil, there was an issue transporting the RNA back to the US for sequencing. And so we unfortunately lost that. So I'm looking forward to coming back to one of the things that Bruno wanted to study was the Mitsuda reaction. And unfortunately, those specimens didn't process correctly. So let's see, there are some questions already. Do I see differences in the pattern of cells when comparing reversal reaction in possible bacillary and multibacillary patients? Well, we just started on the spatial seq and I thought the spatial seq, the reversal reaction was strikingly similar to the tuberculoid. So I felt comfortable with N equals 2 for submitting this paper. But as far as the single cell RNA-seq, so there were I'll go back to sharing. Yeah, I didn't include the table with the classification, but I just want to go back to one slide early on in reference to that. Wow, that's a long time. Yeah, so So just judging by the number of bacilli, there was maybe one that was different, and I really couldn't feel that we detected, comparing 4 to 1, it wasn't that different. I don't think we've done enough samples to know yet. And I'm finally ready to take on borderline lepromatous leprosy, which I've sort of avoided my entire career because I've been confused by why borderline lepromatous leprosy has so many lymphocytes. And I'm to the belief that I wonder if those patients have a low-grade reversal reaction, actually. But I think it will be interesting to apply this to across the spectrum. And so I can't wait. We can't wait to come back and work with you. I think there was another, Dr. Moraes has a question, it says. But it's not, there's no question. But somebody won't.
I do. Hey, Robert, it's
Milton. Yeah, hi. Hi, Milton.
Yeah, good to hear. I hope you don't mind us sneaking in your lecture. It's wonderful so far. It's really amazing data and very clear. Thank you very much. So I have a small comment, and then I would like you to answer one question. Is actually you showed very well that leprosy is a hallmark of especially when we analyze reversal reactions of this kind of granulomatous formation, epithelioid transformation that we see in activation, and how and which are the cells actually participating in this, and the signature that we evaluate. Obviously, there's a lot of overlaps, and but I like it very much a paper from Lenk, I guess it's a Nature Immunology 2018. When they evaluate pulmonary tuberculosis and pulmonary sarcoidosis, and then obviously, as I said, there's some overlap here, right? But we see a lot of genes associated with mTOR pathway and autophagy as an outcome, right? And sometimes, I mean, if you actually block, they have a knockout mice for those tumor suppressor complexes that are regulating mTOR, and they show clearly that actually when you have one of those genes knocked out, you have a kind of overstimulation of the mTOR pathway, and that leads to autophagy, which is amazing. And obviously that I see many of those genes in your antimicrobial signature, but if you recollect or retrieve data, specifically on autophagy and mTOR, did you test it, or because you already have the data sets, right? So it would be very, very interesting to combine this two kinds of analysis. Obviously, as you mentioned, it takes a village to kill the bacteria, and so there are it's a lot of cells working together. But this role in autophagy, is very important as well.
No, we agree about the importance in autophagy, I don't. Well, I think I would call on Rosanny to see if we look at a curated set of autophagy genes, or whether she also did canonical pathway analysis. I don't remember offhand. I don't know. Are you still here, Rosanny?
Yeah, I'm here. Hi, okay. Milton. So we look some autophagy genes, not that much the mTOR pathway, because I'm looking them more in the metabolic pathway, but we look some autophagy genes, and it's kind of mixed, especially when you go through the different myeloid clusters, the macrophage clusters. So usually it's like a little bit spread in the pseudo time, that might be normal, since that you have a lot of post-transcriptional regulation in autophagy, but you have some main things that looks to be up in the reversal side, but we didn't see until I need to double check something different in the emitter expression by itself. But we have some of the canonical autophagy pathway, a little bit up in RR, but not enough to have like a real autophagy pathway when you went to ingenuity or something like that.
But I don't think we looked in the spatial data.
No, we didn't look at yet.
Because I think the spatial data, we pretty much probed for the antimicrobial genes, and I can't recall even, and it's only 2 months ago, whether Fei-Young gave us the list of genes for each cluster. We never did we never did canonical pathway analysis on that. So we should do that. Actually, I should talk to Fei-Young.
Yeah, but that's more clear in the book. In the book RNA-seq. But I remember that I saw something in the single cell really in the beginning, but not really enough to have a clear pathway.
Well, we'll revisit that. We will. That's obviously we agree an important pathway.
Yeah, and the other thing, I mean, if I may, just to complement on that, because if we take a data on genetic analysis on leprosy, right? Most of the genes that now we are actually associated with, especially susceptibility towards the reversal reaction phenotype, just reversal reaction outcome, right? And not talking about leprosy per se. And then we see Parkin, we see LARC, we see a lot of those genes that are, clearly associated, one, with Parkinson's diseases and Crohn's diseases, which are, again, inflammatory diseases that are, we have a lot of granuloma formation and so on. So I would, I'm sorry, I believe that is very, highly convergent, right? I mean, if you take all of these data together, I understand that it's, somehow, obviously in leprosy, we have the antigen trigger or the bacteria, the viable bacteria triggering this kind of response. And it is interesting that we see the same kind of pathways being activated during Crohn's disease or ulcerative colitis. Right. Yeah, and Parkinson's diseases. And well, so it's very nice to correlate all of that, right?
So actually, Priscilla is working with someone at UCLA doing something called mergeomics, where she's taking the published GWAS data for leprosy and combining it with bulk RNA-seq and hopefully down the road single cell and the spatial data to look for how polymorphisms may affect certain pathways. But is there a list of genes that have been found altered in mycobacterial disease somewhere? Because we could merge that list with the pathways as well.
We do have actually we just published its work from Tiago's PhD thesis. We just published its meta-analysis of all the microarray data that we have so far. We cannot merge with RNA-seq data yet because there's some technical issues on how to normalize that. But for the microarray analysis, especially for the data sets that we were able to retrieve the data on, for example, clinical polarized forms for leprosy, right? LL versus BTs. We have a list, and the data sets are also organized. So I mean, we could even share with you if you want.
Yeah, I'd be happy to collaborate on that. It would be wonderful.
Great. That would be nice. Very nice. Great.
And I think that the mergeomics is working pretty well. One of the problems is there isn't an expression table for macrophages, so we're missing some genes, but so far it looks like it's possible to identify some pathways in macrophages that are connected to the GWAS data which is from China, obviously. So I was supposed to go to China and work on the collaboration, but that didn't happen. Okay, question is, let's see, these TREM2 macrophages, foam cells, are cells that hold many scavenger receptors, right? As well as receptors in the iron pathway. Do you see anything in these analyses or some kind of specific macrophage? How am I going to, okay. Hold on 1 second, and I will treat you to, you can look at the data with me. This might scare you though. Okay, well, I showed you part of the spreadsheet for the T cells, which was kind of neatly organized. Now I'm curious if, yeah, granzyme B was down lower. Okay, but this is, the macrophage one is kind of a mess. So this is what I go through when we get data, and I'm trying to figure out what's going on. And so this, oh, here was mTOR signaling somewhere. That was in the dendritic cell. Look at that. Okay, anyway, back to this. You get easily distracted here. Wait, where is this? Oh yeah. Okay, so this is I collect information to try to figure out what's going on. So the TREM2 macrophage at this point was, which one was it now? At one. Oh yeah, I called it an IL-4, IL-10 macrophage, which is interesting because IL-4 can induce a TREM2 macrophage. So those are the genes. This is the kind of list you get. I had no idea what this was for a year. It had ApoE. Now, so MSR1 is a scavenger receptor. There's TREM2. See, at this point, I hadn't even highlighted it. I didn't know what that meant. CD163. So I'm happy to send you the list for you to look at. And also I have a consensus list, various consensus lists. So this is in various papers and oh yeah, so this is like there were 7 papers published, and this is these are the ones that were in 4 out of the 7 papers. So anyway, if you write to me, I'm happy to share any of the information with you. Okay, so I guess without any questions, I thank you again. Oh wait, did you observe exhausted cells in LL versus RR patients? Yeah, I don't know if I could call them exhausted. I did see these cells that looked like they were stressed, and some of this may be due to the processing of the tissue. So I'm not really sure. There are a lot of, I mean, that gets to the question of what haven't I expected to find, or what was different. And one was the T-RELs that made IL-4, but IL-4 is detected poorly. I was surprised by the Th17 cells, but as mentioned, the detail on them is at least IL-26 was there, but not, it was hard to detect IL-17. I was clearly surprised by the populations of fibroblasts. I had no idea about that. And it's very intriguing what role they may play. It was hard to differentiate gamma delta cells from alpha beta tricytotoxics and NK cells. So we need more resolution for that. And I think disappointed from the spatial seq, I couldn't identify Langerhans cells like cells in the dermis yet. So I don't know. Those are the kind of things. Interesting to see LAMP1 in the macrophages in Leprosy. So let's look at that. Because we always have to. So when you make a statement like that, what we have to do is search for LAMP1. Okay, that's in. Oh, so it was both in the TREM2. It's in these things, whatever. We'll get to this. Oh, there it is. That is in. So this is what we do. It's an M3. I don't know why I think it's in TREM2 also, but it's in the transitional ones. Why am I not finding it here? This is what I sit around doing. So I don't know, it must have been, it says that it's in both, but I don't know where you saw that, but it seems to be in the transitional macrophages. Okay, I don't know. I've lost the chat. No. I have to look at the expression of it. That is interesting. Okay. Well, I'm going to move on to my lab journal club. I thank you again, and I hope to see you in person soon.
Thank you very much for this wonderful presentation again, and thank you for being with us today again in another course. Thank you very much.
Okay, my pleasure. Thank you so much.
Thank you very much.
Thank you
Dr.
very
Modling.
much, Robert. Good to see you
Okay,
again.
good to see you. Next time in person. Okay.
Yeah, let's do it. Hope so.
Okay. Bye.
Bye-bye. Thank you very much. Bye.
Bye.
Bye-bye.